Hello, my AP Biology minions. Today we're going to talk about what happens post-transcription, how we get from that RNA strand that was transcribed from your DNA template into something that can be used to generate proteins. So during the process of transcription, the process creates the beginning of messenger RNA. It actually doesn't create messenger RNA. It makes something that will later become mRNA. That something we call a primary transcript, or pre-mRNA. And you can see from this diagram that pre-mRNA has a lot of differences between it and mRNA. You can see above everything else, even this right here is longer than the mRNA because there are aspects of this that are not in mRNA. And even pieces of this section right here, this section right here, are not part of the transcription process. So let's talk about what happens. So pre-mRNA is generated in the process of transcription. Immediately post-transcription, this messenger RNA strand is modified. So the first thing that happens is that a strand of guanine is added to the 5' end, and that's right here. Okay, so right here, a piece of guanine is added to the messenger, the pre-messenger RNA. It's added to the 5' end, and we call it the 5' cap. Now, it has some uses later on after the pre-mRNA has been modified. This 5' cap will be used so a ribosome can recognize the messenger RNA in the process of translation. On the other end, all right, so down here, right here, you've got this structure, which is actually a string of adenine nucleotides that is attached to the end. And these adenine nucleotides get called a poly-A tail because it's multiple A's, multiple adenines. And the poly-A tail actually helps the cell get the messenger RNA out of the nucleus. Again, transcription, the process that generates this primary transcript or this pre-mRNA, occurs in the nucleus. This modification that we're talking about in this presentation happens in the nucleus. But then the messenger RNA needs to be removed from the nucleus because the next step happens outside the nucleus. So this poly-A tail right here, this actually helps get the messenger RNA out of the nucleus so that it can be used. In addition to their individual uses, the 5' cap aids in ribosomal recognition and the poly-A tail adds in transport out of nucleus. Both of these additions to the primary transcript, to the pre-mRNA, actually aid in making sure that enzymes don't digest pieces of the pre-mRNA transcript. As you know, if you change it, then you're going to change the structure of a protein, which could be a problem. All right. So this process sort of continues, hence my reuse of the previous diagram, all right? To understand how it continues, all right, so we've got our first thing where this 5' cap has been added right here, the guanine that's used for ribosomal recognition. And then down here we've had this poly-A tail added so that we can get this sucker out of the nucleus when we're done with it. But then we've got to talk about everything that's inside those two things, so all the stuff that fits in this gap right here. And what you have to understand is that there's actually two different sections in that run right there. Some of them are called introns, and some of them are called exons. Introns are segments of DNA, like this, that are not coded. So these segments of DNA that get transcribed are not used to generate proteins. They are non-coding. But the exons, all right, these sections right here are coded. They are used to generate a protein. So what happens is this pre-mRNA has to be spliced. It has to have the introns removed and the exons added together, like combined, so that they create one unified string. So each exon has to be sort of excised out and recombined and then the introns are removed because they're non-coding. All right. So what happens is on either side of the intron, so like if this is our intron, on this side right here and on this side right here are small sections of non-coding nucleotides. And it is those nucleotide sequences that are the signals for the structure, which is known as a spliceosome. All right, this structure right here is what does this removal, this excision of the introns. Okay, these non-coding regions on either side of the intron are the key that lets the spliceosome know when to do its job. Now, the spliceosome is actually a complex of structures. It's actually something we call small nuclear ribonucleoproteins, all right? So they're basically just, small pieces of proteins that are associated with RNA, and then also small pieces of RNA that join together. Remember, we talked earlier about how RNA has some enzymatic properties. So these protein structures and this RNA structure allow this excision, this splicing of exons to occur. And so we call the combination of nucleoproteins and nuclear RNA a spliceosome. And the spliceosome actually removes the introns and joins the exons together, and that creates what we finally would call messenger RNA. Now, messenger RNA isn't actually done after that, all right? We talked about the ribozyme activity. We talked about removing the introns and the exons. And one of the things you have to know is that this process of splicing can actually help regulate how much mRNA or how quickly mRNA gets out of the nucleus. And if you think about it, how much mRNA is removed or how quickly the mRNA gets out of the nucleus can determine how quickly or how much protein is generated in a cell. So that's a way for the cell to control the amount of chemicals it produces, which is a way for the cell to control what it's doing. Now, if you take a look over here, right, you've got your transcript up here, all right? So this green is what's been transcribed, right here, what I'm circling, is what's been transcribed by the process of transcription. So transcription pops this out of the DNA, and we've got our pre-mRNA, our primary transcript. You'll notice the RNA cap, the 5' cap, the guanine, has been added. That way ribosomes will be able to recognize this later. Okay, so here we've got our ribosome section. And then over here we've got our poly A tail, and that's for transport out of the nucleus. All right, so we've done that editing already. Those pieces are immediately added. And then what happens is you've got these pieces right here, these small purplish circles. They are the small nuclear ribonucleoproteins and the small nuclear RNA that combine to drag this intron out of the space between these exons. This is a spliceosome complex. Then this exon, these two exons have been combined to create what is right here, our mRNA. So this right here is what we call mRNA. And the mRNA is then transported out of the nucleus by structures that are in the nuclear membrane. And the signal for that transport comes from the poly A tail. Okay? Now, the thing about introns, if you think about it, like literally like 30 to 50% of your DNA is non-coding introns, which seems almost a waste. But there's a lot that goes into that. Introns can be used to regulate gene activity in a lot of ways. Sometimes introns are used for promoter regions of other genes. Sometimes they're used for allowing a different way of splicing. All right. Sometimes if introns are removed in different sequences or in different amounts, this can create different combinations of exons. Sometimes the structure of introns and their positioning on a DNA strand allows places for crossing over to occur. Because if you think about it, you don't want crossing over to happen in the middle of an exon. Not often. But if you have crossing over happen in an intron, who cares if there's a couple of mistakes? It's not being coded anyway. Okay? And then sometimes what happens is these exon structures are actually for general domains inside a protein. And so you could sort of recombine the domains in different ways. You could actually take those domains from one allele and shift them to another allele. Because remember, you have two alleles for every trait, and your traits are your genes. So you could actually pop the exons back and forth, and that can create variety. Or you could actually have exons pop around across genes during this process, which can create variety. Remember, variety is not always good or bad. It could be both. All right? It's just that if the potential for creating variety does not exist, then evolution will not occur, and organisms will not be able to survive changes in their environment. All right. I think that's all we've got for how we edit RNA post-transcription. So we've gone from transcription into pre-mRNA and primary transcripts. And in this presentation, we've discussed how pre-mRNA and primary transcripts are altered so that they can become messenger RNA, which is removed from the nucleus and will be used in our next protein synthesis process.